Chapter 1, Cellular and Molecular Biology of Aqueous Humour Dynamics, page number 15, from Shields Textbook of Glaucoma, 6th edition, Part 5. Biology of Aqueous Humour Outflow. As noted earlier, most of the aqueous humour leaves the eye at the anterior chamber angle through the system consisting of trabecular meshwork, the Schlemm canal, intrascleral channels, and episcleral and conjunctival veins. This pathway is referred to as the conventional or trabecular outflow. In the unconventional or uveoscleral outflow, aqueous humour exits by passing through the root of the iris, between the ciliary muscle bundles, then through the suprachoroidal scleral tissues. The relative contribution of these outflow pathways depends on the species studied. Furthermore, there is an age-dependent change in aqueous humour outflow in both the trabecular and uveoscleral pathways. In general, the trabecular outflow in human eyes accounts for approximately 70% to 95% of the aqueous humour egress from the eye, with the lower values corresponding to younger eyes and the higher values corresponding to older eyes. The other 5% to 30% of the aqueous humour leaves primarily by the uveoscleral outflow pathway, with a decline in the contribution of this pathway with age, whereas both total outflow facility and trabecular outflow facility also decline with age. The relative contributions of trabecular and uveoscleral outflow show an age-related shift, with a relative increase in the contribution in the trabecular pathway, because uveoscleral outflow is relatively independent of intraocular pressure in the physiologic range. Decreased uveoscleral outflow and increased trabecular outflow resistance with age simply mean that intraocular pressure must increase sufficiently to drive a higher proportion of total flow, which remains rather constant with age across the increased trabecular resistance. Cellular organization of the trabecular outflow pathway. Scleral spur. The posterior wall of the scleral sulcus is formed by a group of fibers, the scleral roll, which run parallel to the limbus and project inward to form the scleral spur, which is composed of 75% to 80% of collagen and 5% of elastic tissue. Myofibroblast-like scleral spur cells, in close association with varicose axons characteristic of mechanoreceptor nerve endings, suggest there is a mechanism for measuring stress or strain in the scleral spur, as might occur with ciliary muscle contraction or changes in IOP. Schwalbe line. Just anterior to the apical portion of the trabecular meshwork is a smooth area which varies in width from 50 to 150 micrometer and has been called zone S. The anterior border of the zone consists of the transition from trabecular to corneal endothelium and the thinning and termination of the Descemet's membrane. The posterior border is demarcated by discontinuous elevation called the Schwalbe line, which appears to be formed by the oblique insertion of uveal trabeculae into limbal stroma. Clusters of secretory cells called Schwalbe line cells have been observed just beneath this ridge in monkey eyes and are believed to produce a phospholipid material that facilitates aqueous humour flow through the canalicular system. Trabecular meshwork. As previously discussed, the scleral sulcus is converted into a circular channel called the Schlemm's canal by the trabecular meshwork. This tissue consists of a connective tissue core surrounded by endothelium and may be divided into three portions: A. Uveal meshwork, B. Corneoscleral meshwork, and C. Juxtacanalicular tissue, which is sometimes referred to as the cribriform layer. Uveal meshwork. This innermost portion is adjacent to the aqueous humor in the anterior chamber and is arranged in bands or rope-like trabeculae that extend from the iris root and ciliary body to the peripheral cornea. The arrangement of the trabecular bands creates irregular openings that vary in size from 25 to 75 micrometer across. Corneoscleral meshwork. This portion extends from the scleral spur to the anterior wall of the scleral sulcus and consists of sheets of trabeculae that are perforated by elliptical openings. These holes become progressively smaller as the trabecular sheets approach the Schlemm's canal with a diameter range of 5 to 50 micrometer. The anterior tendons of the longitudinal ciliary muscle fibers insert on the scleral spur and posterior portion of the corneoscleral meshwork. This anatomic arrangement suggests an important mechanical role for the cholinergic innervation of ciliary muscle on trabecular meshwork function. Both the uveal and corneoscleral trabecular bands or sheets are composed of four concentric layers. First, an inner connective tissue core is composed of typical collagen fibers with the usual 640 angstrom periodicity. Indirect immunofluorescence studies of human trabecular meshwork indicate that the central core contains collagen types 1 and 3 and elastin. Second, elastic fibers are composed of otherwise typical collagen arranged in a spiraling pattern with an apparent periodicity of 1000 angstrom. These spiral fibrils may wind loosely or tightly and may provide flexibility to the trabeculae. Third, glass membrane is a name given to the layer between the spiraling collagen and the basement membrane of the endothelium. It is a broad zone composed of delicate filaments embedded in a ground substance. Fourth, an outer endothelial layer provides a continuous covering over the trabeculae. The trabecular endothelial cells are larger, are more irregular, and have less prominent borders than corneal endothelial cells. They are joined by gap junctions and desmosomes, which provide stability but allow aqueous humor to freely traverse the patent endothelial clefts. Two types of microfilaments have been found in the cytoplasm of human trabecular endothelium. 60 angstrom filaments are located primarily in the cell periphery around the nucleus and in cytoplasmic processes. These appear to be actin filaments, which are involved in cell contraction and motility, phagocytosis, pinocytosis, and cell adhesion. Intermediate filaments of 100 angstrom are more numerous in the cells and are composed of vimentin and desmin, according to immunocytochemical studies of cultured human trabecular cells. These molecular markers in the trabecular endothelial cells suggest a myocyte or muscle cell-like phenotype, which further implies important contractile and motility functions. Juxtacanalicular tissue. This portion of the trabecular meshwork differs histologically from the other parts of the meshwork and has been given various names including juxtacanalicular connective tissue, pore tissue, cribriform layer, and endothelial meshwork, depending on how one defines the anatomic limits of the tissue. In the broadest sense, this structure has three layers, discussed here beginning with the innermost portion. The inner trabecular endothelial layer is continuous with the endothelium of the corneoscleral meshwork and might be considered as a part of this layer. The central connective tissue layer has variable thickness and is unfenestrated with several layers of parallel spindle-shaped cells loosely arranged in a connective tissue ground substance. This tissue contains collagen type 3 but no collagen type 1 or elastin. Connective tissue cells in human and rabbit trabecular meshwork contain coated pits and coated vesicles in the plasma membrane, which are involved in receptor-mediated endocytosis. The outermost portion of the trabecular meshwork, that is, the last tissue that aqueous humor must traverse before entering the canal, is the inner wall endothelium of Schlimm's canal. This endothelial layer has significant morphologic characteristics which distinguish it from the rest of the endothelium in both the trabecular meshwork and in the Schlimm canal. The surface is bumpy due to protruding nuclei, cyst-like vacuoles, and finger-like projections bulging into the canal. The finger-like projections have been described as endothelial tubules with patent lumens, although there is lack of agreement as to whether they communicate between the anterior chamber and Schlimm canal. Actin filament, as previously described in the uveal and corneoscleral trabecular endothelium, are also present in the inner wall endothelium of Schlimm's canal. The intercellular spaces are 150 to 200 angstrom wide, and the adjacent cells are connected by various intercellular junctions. It is not clear as to how tightly these junctions maintain the intercellular connections, although they will open to permit the passage of red blood cells. Zonal occlusions have been demonstrated in primate studies, which are traversed by meandering channels of extracellular space or slit pores, although it is estimated that this accounts for only a small fraction of the aqueous humor that leaves the eye by the conventional route. Openings in the inner wall endothelium of the Schlimm's canal have been described, and in general, the openings consist of minute pores and giant vacuoles that vary in size, ranging from 0.5 to 2 micrometer. Evidence in support of their role in the transcellular outflow is based on injection of tracer elements into the anterior chamber, with demonstration of the tracers in the vacuoles and pores. The observation that the concentration of tracer material in the giant vacuoles is not always the same as in the juxtacanalular connective tissue, such as a dynamic system in which the vacuoles intermittently open and close to transport aqueous humor from the juxtacanalular tissue to the Schlimm canal. This transcellular transport has active and passive mechanisms. Indirect evidence for active transport includes the demonstration of enzymes and microscopic structures compatible with an active transport system in or near the endothelial layer. However, the bulk of evidence supports the theory of passive pressure-dependent transport because the number and size of the vacuoles increase with progressive elevation of IOP. It has been proposed that potential transcellular spaces exist in the inner wall endothelium of the Schlimm canal, which open as a system of vacuoles and pores, primarily in response to pressure to transport aqueous humor from the juxtacanalular connective tissue to the Schlimm canal. If intracellular transport to the inner wall of endothelium of the Schlimm canal exists, it has been calculated on the basis of the estimated size and total number of pores and giant vacuoles that resistance to outflow to this system accounts for only a small fraction of the total resistance to aqueous humor outflow. It is also possible that only a portion of the juxtacanalular tissue actually filters. It has been suggested that aqueous humor flows preferentially to those regions of the juxtacanalular connective tissue nearest the inner wall pores, creating a funneling effect, which increases apparent flow resistance in the connective tissue by approximately 30-fold. An alternative theory to that of transcellular transport is paracellular routes between the inner wall endothelial cells. Perfusion of monkey eyes with cationized ferritin reveals separation of adjacent cell membranes between tight junctions forming openings and tunnel-like channels, which stained with the tracer indicating intercellular passage. These paracellular pathways were larger at higher perfusion pressure, and apparent giant vacuoles were often dilatations of the paracellular spaces. Of historical interest, the Sondermann Canal, although originally described as endothelial line channels communicating between the Schlemm Canal and intertrabecular spaces, have subsequently been interpreted as tortuous communications wandering irregularly and obliquely through the meshwork. Schlemm Canal. This 360-degree endothelial lined channel averages 190 to 370 mm in diameter, that is 1.9 to 3.7 cm in diameter, with occasionally branching into a plexus-like system. The endothelium of the outer wall is a single cell layer that is continuous with the inner wall endothelium but has a smoother surface with larger, less numerous cells and no pores. The outer wall also differs in having numerous large outlet channels, which are described below. Smooth muscle myosin containing cells have been localized in the human aqueous humor outflow pathway adjacent to the collector channels, slightly distal to the outer wall of Schlemm's canal. Torus or lip-like thickenings have been observed around the openings of outlet channels, and septa have been noted to extend from these openings to the inner wall of Schlemm canal, which presumably help keep the canal open. The endothelium is separated from the collagenous bundles of the limbus by a basement membrane and fibroblasts. Episcleral and conjunctival veins. The Schlemm canal is connected to episcleral and conjunctival veins by a complex system of intrascleral channels, the aqueous veins of Escher, which are now more commonly referred to as collector channels, have been defined as originating at the outer wall of the Schlemm canal and terminating in episcleral and conjunctival veins in a lamination of aqueous humor and blood, referred to as the laminated vein of Goldman. Two systems of intrascleral channels have been identified. A direct system of large caliber vessels which run a short intrascleral course and drain directly into the episcleral venous system, and B. An indirect system of more numerous final channels which form an intrascleral plexus before eventually draining into the episcleral venous system. The intrascleral aqueous channels do not connect with vessels of the uveal system except for occasional fine communications with ciliary muscle. The aqueous vessels join the episcleral and conjunctival venous system by several routes. Most aqueous vessels are dilated posteriorly and drain into episcleral veins, whereas a few cross the subconjunctival tissue and drain into conjunctival veins. Some aqueous vessels proceed anteriorly to the limbus, with most running a short course parallel to the limbus before turning posteriorly to conjunctival veins. Casting studies in rabbit and dog eye revealed a wide venous plexus in the limbic region of the episcleral vasculature anastomosing with a small arteriolar segment, the latter of which contains smooth muscle cells that may have a role in regulating aqueous humor drainage by the episcleral venous plexus and subsequently influencing the intraocular pressure. In the rhesus monkey, the conjunctival vessels receiving aqueous humor drainage have a diameter consistent with that of capillaries, whereas most of the vessels in the episcleral plexus are the size of venules. Both types of vessels have simple walls composed of endothelium and a discontinuous layer of pericytes through which tracer element, for example, horseradish peroxidase, and presumably aqueous humor freely diffuse into subconjunctival and episcleral loose connective tissue. The episcleral veins drain into cavernous sinus via the anterior ciliary and superior ophthalmic veins. whereas the conjunctival veins drain into superior ophthalmic or facial veins via the palpebral and angular veins. Cellular organization of the uveoscleral pathway. The unconventional outflow for aqueous humor outflow has not been studied as extensively as the trabecular outflow pathway. Historically, two unconventional pathways have been discriminated. A. Through the anterior uvea and the iris root, which is referred to as the uveoscleral pathway, through transfer of fluid into the iris vessels and vortex veins, which has been described as uveo-vortex outflow. Uveoscleral outflow. Tracer studies have shown that aqueous humor passes through the root of the iris and interstitial spaces of the ciliary muscle to reach the suprachoroidal space. From there, it passes to episcleral tissue via scleral pores surrounding the ciliary blood vessels and nerves, bases of optic nerve membranes, or directly through the collagen substance of the sclera. Studies with cynomolgus monkeys revealed a lower hydrostatic pressure in the suprachoroidal space than in the anterior chamber, and it was suggested that this pressure differential is the driving force for uveoscleral outflow. The extracellular matrix of normal human ciliary muscle contains collagen 1, 3, and 4, fibronectin, and laminin in association with muscle fibers and blood vessels, and it has been suggested that the biosynthesis and turnover of these glycoproteins may play an important role in resistance to flow within the unconventional pathways and in mediating the action of certain pharmacological agents. This is discussed further in the following section on molecular mechanisms of outflow resistance and in chapter 28 on prostaglandins. Uveo-vortex outflow. Tracer studies in primates have also demonstrated unidirectional flow into the lumen of iris vessels by vascular transport, which is not energy dependent. The tracer can penetrate vessels of the iris, ciliary muscle, and anterior choroid to eventually reach the vortex veins. However, the role of net fluid movement into the iris vasculature is probably clinically insignificant. Some evidence suggests that there is a process of net osmotic resorption of some aqueous humor into the uveal venous circulation driven by the high protein content in the blood vessel in these vessels. The relative contribution for this fluid outflow pathway is not understood for the healthy eye, but it may be clinically relevant in an eye with nanophthalmos. Figure 1.9, light microscopic view of the Schleim's canal SC and adjacent trabecular meshwork TM of normotensive rhesus monkey eye, trabecular wall of the Schleim canal TW with prominent vacuolated cells, arrows, corneoscleral wall of Schleim's canal CW, collector channel CC, toluidine blue stain, 1030 times magnification. Figure B, 1.9, Figure B, electron microscopic view of trabecular walls of SC of normotensive human eye showing vacuolated endothelial cells V containing flocculent material FL, OZ, occluding zonules, BM, basement membrane, OS, open spaces in endothelial meshwork, 15,000 times magnification. Figure 1.10, schematic of aqueous humor outflow distal or beyond the conventional or trabecular pathway and into the canal of Schleim. The canal divides into two or more portions intermittently. The drawing is divided into four portions by the dotted lines. The internal connector channels of Sondermann are labeled in the upper right sector as they extend into the trabecular meshwork. The external corrector channel are seen in the upper and lower right sectors, arising from the canal and uniting with the deep intrascleral plexus of extending directly to the episcleral veins. The deep and intrascleral venous plexuses are external to the canal. In the upper left sector, an aqueous vein arises from the deep scleral plexus and another arises from the Schlemm canal and runs directly to the episcleral venous plexus. External collector veins are seen to arise from the canal and join the deep scleral plexus. In the lower left sector, the arteries of the deep sclera are seen to be in close relation to the canal of Schlemm. Thank you.